This is part 3 of Chapter 14's lecture on Mendel's genetics. And here we have a Punnett square. So this should be very familiar and a lot of review for you, but just want to make sure that we are familiar with how to use these. So on a very basic level, we have this Punnett square where we're crossing a heterozygous male and a heterozygous female. And typically what we do with a Punnett square is we put the male across the top and the female across the bottom. So the alleles here of the male are found here, which represent the sperm, that gamete. And then the females, again, that was heterozygous, along the side here, which represents the female egg. And then we just add these together for each of the blocks here. So here we have two dominant P's, or for purple here, ending up with homozygous dominant, and so on. So you fill in your square. And what you end up seeing from this is out of the 4 offspring that you can get by combining these two sets of alleles, we end up with this ratio. So again, this is our common 3-to-1 phenotype. The phenotype of 75% or 3-to-1 versus 25% to the white. But when we look at the genotype, we see something different. What we see in terms of the ratio is a 1 to 2-to-1 ratio typically, where only 1 or 25% proportionally are going to be homozygous dominant. But we're going to end up with half of the offspring becoming heterozygous, and then only 1/4 would end up being homozygous recessive. So that's our 1 to 2 to 1. So remember that genotype is the description of the alleles, the specific alleles that are actually determining what the phenotype, what the expression is of the character. And if it's homozygous, that means we have the same alleles, whether either dominant, capital P's, or recessive, lowercase p's, or we have heterozygous for different alleles. And then 2 organisms can have the same phenotype, but they can have different genotypes. So the only time we have this is when we have the dominant trait being expressed. So in the case of flower color, when we have purple being expressed, we can have 2 different genotypes that will express the phenotype of purple. It could be homozygous dominant with 2 capital P's, or it can be heterozygous with a capital P and little p. And so if we are looking at F1 or F2 generation and we see the dominant trait being expressed, we really don't know which of these genotypes it has. So how do you determine if that individual has either homozygous or heterozygous alleles? And the way we do that is with a test cross. So a test cross is simply breeding the dominant phenotype, that is the unknown, which would be our 2 capital P's, or our heterozygous capital P, lowercase p. And so this is our question mark, and we cross that with a homozygous recessive individual. And what that will tell us will be here. So what that's going to tell us is if it is the dominant P's here, so if it's homozygous dominant, what we would end up with an entirely hybrid set of offspring in the F1. So when we're crossing that again with the homozygous recessive, this is our test, and this is our known, because we know if something is showing a recessive trait, it's homozygous recessive. So we can put that here. We know these. This is our unknown. And we can see that if we end up with this, then we started with a homozygous dominant. But if it was heterozygous, what we're going to end up with by crossing it with a homozygous recessive is half of them showing the dominant trait and half of them showing the recessive trait. So this is how you can tell again whether your phenotypic dominant trait is being coded by alleles that are heterozygous or homozygous. So when we're just looking at one trait, what we're looking at is a monohybrid cross. And Mendel's first experiments did only follow a single character like flower color. And then all the F1 progeny produced in these crosses were called monohybrids. They were, in this case, heterozygous for one character. When we have a cross between two heterozygotes, that's called a monohybrid cross. On the other hand, if we have a dihybrid cross, we actually are looking at two character traits. So for example, seed color and seed shape. So we'd have to have two different characters, and it was only by doing this that Mendel was able to study and identify the second law of inheritance, because he needed to see how two different characters actually ended up assorting themselves throughout meiosis in the formation of gametes. And he was able to do this by looking at different character traits. Fortunately, he was able to choose character traits that were actually found on different chromosomes, so he was able to identify that they were assorting independently. But this is an example here of a dihybrid cross, and in this case we have true breeding. Again, the parent generation is always going to be true breeding, which means it's homozygous for the dominant trait versus homozygous for the recessive traits. And a true breeding with two traits, two characters, one's yellow and one's round. So it's going to be color is the character and shape of the peas is the other character. And then the recessive traits are green and wrinkled. So you can see here the legend or key here showing you what we're looking at. And what was found with the dihybrid cross is, again, an F1 where 100% of the offspring showed the dominant trait, which we now know, of course, made them all heterozygotes. So the dominant trait showed up again, and the recessive trait totally disappeared in the F1. But then when we have the next generation, the F2, by crossing the F1s, we had our recessive traits being wrinkled and green reappear. But now, because we have a couple different traits that we're looking at, we actually end up with a different ratio, because we ended up with some that showed just the recessive trait in one thing, such as shape. So here we have, actually this one's showing just the color and the shape being round. So it had the dominant shape, but it had the recessive color. Or in this case, we had the dominant color, but the recessive shape, so it's wrinkled. So these two middle ones are the ones that ended up with a mix of dominant and recessive. And the ratio in the case of this type of dihybrid cross is typically and traditionally a 9:3 to 3:1. So we end up with 9 showing this dominant yellow round peas. We have a 3 out of the 16 showing green round peas, 3 showing yellow wrinkled peas, and only 1 showing the parental green wrinkled peas. One thing to note that's kind of interesting about this experiment is that the wrinkled seeds in pea plants with 2 copies of the recessive allele end up being wrinkled because of an accumulation of monosaccharides and excess water in the seeds because of the lack of a key enzyme. The seeds wrinkle when they dry up. So both homozygous dominants and heterozygotes are able to produce enough of the enzyme to convert all of the monosaccharides into starch, and that forms a smooth seed when they dry. But it's the lack of this enzyme which causes the wrinkling to occur. So I thought that's kind of interesting, and to help you start thinking in terms of what does it mean to have the recessive trait? Because oftentimes the recessive trait means that something is not being made correctly, or either something is malfunctioning or there's a total absence of a needed protein, in this case an enzyme, to help with or determine whether we have monosaccharides being converted into starch or not. Okay, so if the genes are on different chromosomes, how do they assort in the gametes? Do they do it together or do they do it independently? So we could either have it where these two end up in one and these two end up in another, so this way and this way, or we could have it looking like this. And as you know, since this is a review, that what we actually end up with is this here. So again, here is if you were to make a Punnett square for a dihybrid cross, and practice doing this if you haven't, because it does help you to see how things actually work out. But I think the biggest advantage to doing the Punnett square for a dihybrid is that you practice making the potential gametes, because what you're lining up here and here are the possible gametes, because remember, we're only going to get one allele from each homozygous pair. So these two Ys are homologous chromosomes is what I meant to say, and these two are homologous chromosomes. And when we make a gamete, the gamete's only going to have one of these homologous pair. And so that's how we end up with just two alleles in the gamete, because here we're going from diploid of 4 to haploid of 2. So remember that, because how you set this up is really the only tricky part, because once you do, it's quite easy to fill in the rest and figure out what is the genotype and phenotype. So this is talking about probability, but I'm actually going to come back to this at the end of this lecture. So let's just keep on going here. Okay, so this is just a summary of looking at the chromosomal basis of Mendel's laws. So if we look at this again, we're going to see how we have our actual chromosomes going through meiosis. So when we have our gametes, so through meiosis we have formed gametes, which means that we're going to end up with fertilization of a zygote right here. And so this zygote is again the F1 generation, which in this case ended up being heterozygous for both traits. So with the principle of segregation, we end up with our tetrads being lined up here, and we are going to separate out the two allele possibilities for each set of homologs. And that's what happens here in anaphase 1. But what's also occurring through this metaphase-anaphase stage is the principle of independent assortment, which means that some of the father's chromosomes are going to end up in one side of the cell, and some are going to end up in the other, just depending on how they lined up on this metaphase plate. So again, I want you to think about what that means and make sure you understand how that works. So fortunately for Mendel, he chose peas wisely because pea plants are good for genetic research. They're available in a lot of varieties, and they have distinct heritable features with